Hello everyone. So now we are going to start the part 2 from the drug stability. So in this particular presentation we are going to deal with the different mechanism of drug instability, means those mechanisms which involve to make the drug unstable. So without wasting any time, let's start our presentation. So as discussed earlier, there are different types of reactions, right, which actually affects the stability of the drug. So first of all, let's discuss the polymorphism. So what is polymorphism? Polymorphism means a molecule or a compound have the tendency to stay in more than one configuration. So polymorphs actually they show significant differences, and it is really very important because you see these polymorphs, suppose sulfur, you know that the sulfur they have polymorphs, right? This polymorph actually show differences in the properties such as solubility, dissolution rate and melting point. So one form of sulfur may have different solubility, different dissolution rate and different melting point, whereas the other form of sulfur will have different in this three parameter. So generally you see this polymorphic changes the crystal form of the drug, right? And this may cause in the change in the solubility. So once the drug, if it has changed from one polymorph to another polymorph, then what will happen? It will affect the solubility, dissolution rate and the melting point of the drug, and maybe will not get the desired therapeutic efficacy, right? For example, you can consider cortisone acetate suspension. This cortisone actually they have the tendency to show the or form the polymorphs, and this led to the degradation of the drug. So how we are going to prevent the formation of the polymorphs? Simply, if we have the suspension, here we have discussed regarding the cortisone acetate suspension. For suspension, we can use a better quality suspending agent like methyl cellulose and ethyl cellulose to prevent the formation of the polymorph. The next type of degradation, it is the photolysis. See, the name itself will say photo means light, lysis means degradation or breaking. So breakage of a compound with the help of the light. So when a molecule it is exposed to electromagnetic radiation, right? Electromagnetic radiation means the light. So they absorb the photons, that is the energy, at a characteristic wavelength. You know that all the substances in this world have the tendency to absorb the light, but with different wavelength. So when any compound it is exposed to electromagnetic radiation, it may absorb the light, that is the photon, at a particular wavelength, and this led to what? Decomposition of the drug molecule. So for example, you can take sodium nitroprusside in the aqueous solution. Sodium nitroprusside is a drug. It is generally given intravenously for preparation for the prevention of acute hypertension. So sodium nitroprusside, it have the tendency to get degraded upon exposure to light. The next example it is your iodine. So how you can prevent this type of degradation of the drug? Simple, use of amber colored bottle. Means if we use amber color bottle, the reddish type of bottle, this will prevent the light to enter within the bottle. So this can lead to prevention from the photolysis. Moreover, storing the product in the dark. So I hope you all have studied in the instruction, the manufacturer instruction. Most of the time we see keep away from the light, right? So we have to keep the bottle away from the light, that is in the dark. The next thing, it is the color changes. Next form of drug instability, it is the color changes. So you see the color changes actually it indicate what? We have a drug, we have a formulation, and now if the formulation is changing its color, what it actually indicates? It simply indicates the chemical or photochemical decomposition of the active ingredients or the dyes or other ingredients. So the color changes generally it takes place or it's happen in two ways in the pharmaceutical products. First, it is the loss of the color. Suppose we have a color, suppose blue color is there in the formulation. After some time the color may fade out, there is no blue or development of color. this blue color may get converted into violet color, right? So this is the development of a new color. Example, phenolphthalein. It's a general basic indicator. We know that, right? It changes its color when the pH is changes. We all know that phenolphthalein is generally colorless in the acidic solution, but it becomes pink in the basic solution. Similarly, ascorbic acid tablet, it turned yellowish brown color. Ascorbic acid tablet, it turned yellowish brown color. Clear? So how we are going to prevent it? Protect the product from the light and air. Simple. You just protect the product, it is a drug product or formulation, from the exposure from the light and air. And you can avoid using the reducing substance as additives, right? The next type of degradation, it is chemical degradation. So in the chemical degradation, you see we have different part. Hydrolysis. Hydrolysis, we know what is hydrolysis. It is the decomposition of any substance by virtue or by the help of water. Then isomerization, racimization. Then we have the epimerization, decarboxylation, elimination and oxidation. Let's discuss one after another. First, let's see the hydrolysis. So this hydrolysis, it generally happens in this type or this category of bond in the drug or drug molecule which have this type of bond. First of all, ester. Next one, amide. Then we have a class of drug like barbiturates, hydrogens, and amides. Then we have the carbon nitrogen bond cleavage. It also takes place with the presence of the water and it done by generally hydrolysis. Let's have some example. Ester. We all know that what is a ester, right? So you see this ester cleavage, actually it involves the acyl group and acid cleavage, right? In the hydrolysis, the cleavage will take place between the acyl group and the acid, right? For example, it takes place in the aspirin, atropine, physostigmine and procaine. So this is a ester, right? This is a ester. Now cleavage will take place here. When the cleavage will take place here, we are going to get one, what is this, acidic group and one alcoholic group. So here ester bond may get break by the virtue of hydrolysis reaction. The next one, it is the amides. So in amides, you see, the thing is that amides, they are less susceptible to hydrolysis as compared to that of the ester. But yes, they also have the tendency for hydrolysis, but their tendency is less as compared to that of the ester. So the leaving group, the group which will leave after the hydrolysis. Earlier, we got one acid group. In the ester, we got one acid group, one alcohol group. But here, in the cleavage of amides, you see the leaving group, it is a amine. It is the amine, the leaving group, the group which will come out. The group which will come out from the compound, it's a amine. So it's a very poorer leaving group. So it actually, this reaction involves the cleavage of the amide linkage. So let's have one example. Example, chloramphenicol and barbiturate. Here the amide linkage takes place. And how it takes place, you see, this is the amide, this is the water. You see here what will happen, we'll get one acidic group and one amine group. This is the leaving group, it is the amine. As I already told you, the leaving group, it's a amine. It's an amine group, right? So now for drugs like barbiturates, hydrogens and amides, you see these group, the barbiturate, hydrogens and amides, they contain functional group related to amides, and they have a tendency to be more reactive. They have a tendency to become more reactive. So what will happen? These barbiturates, actually it may form barbituric acids. So you see the barbituric acid like barbital, phenobarbital and amobarbital, they actually undergo generally ring opening hydrolysis. Ring opening hydrolysis. I repeat, ring opening hydrolysis. As the barbiturates, which have a ring structure, upon hydrolysis the ring will break, the ring will open. This is the reaction, you see. This is the reaction. Here we have a ring. After hydrolysis, the ring may get open. The ring will break. The structure of the ring will break. Clear? The next one, it is the C and bond cleavage, the carbon nitrogen bond cleavage. If the drug have the carbon nitrogen bond and if the water is present, in such cases this carbon nitrogen bond may get break. For drugs like benzodiazepines, they are sedatives, right? They are used to induce the sleep. So benzodiazepines like diazepam, oxazepam, nitrazepam, this type of drug, they generally undergo ring opening due to hydrolysis of the amide and azomethine bonds. I repeat, the benzodiazepine, they generally undergo ring opening due to the hydrolysis of what? Amide and azomethine bonds. So the benzodiazepinoxazoles. So example, oxazole contains benzodiazepines, such as example is your oxazolam, futazolam, haloxazolam, and then cloxazolam. They generally undergo ring opening due to the hydrolysis. Let's have one example. You see, breakage of the ring, right? They will undergo ring opening. They will undergo ring opening due to the carbon nitrogen breakage. Clear? So prevention of hydrolysis. We have come to know this hydrolysis, it really creates lots of problem for the stability of the drug. How we are going to prevent it? See, we have, first of all, we need to store the product in a moisture resistant packaging, such as strip packaging. So the drug, generally the tablets or the capsules, they are generally available in the strips, right? In the strips they are available. So these strips actually, they prevent the hydrolysis, as because this strip, they prevent the water from entering into the drug or into the tablet, right? Next, the hydrolysis of certain drugs such as benzocaine and procaine, it can be also decreased by addition of specific complexing agent. What type of complexing agent may be used? Caffeine we can use. This caffeine, it will act as a complexing agent to prevent the hydrolysis of the drug like benzocaine and procaine. Clear? The next concept, it is the isomerization. We know what is isomerization. So isomerization, it is a process by which what happens? One molecule is transformed into another molecule which has exactly the same atoms, but these atoms are rearranged. So you see, for example, we have this pilocarpine. It's a drug, right? It's a drug, pilocarpine. This pilocarpine drug may undergo isomerization to form isopilocarpine. You can see the change in the position of the what? Atoms. Rest all same, right? The next one, it is the racimization. So racimization, actually it refers to the conversion of one enantiomer into another enantiomer, right? So what are enantiomers? We know that they are optically active, right? So it involves optically active form of a drug into its enantiomer. The drug has to be optically active. If the drug is optically active, the chances is that the drug may undergo racimization. So racimization, if it happens, then one active form of drug may get converted into its enantiomorph. For example, this is. So you see here, change of the position of the OH, right? It has changed, racimization. The next concept, it is epimerization. So what is epimerization? We all know that, right? It occurs with the compounds having more than one symmetric carbon atom in the molecule. If the molecule it have more than one symmetric carbon atom, the molecule has a tendency to undergo epimerization. So at equilibrium, when this will undergo epimerization, at equilibrium there will be a point, right? There will be a point in the reaction. At equilibrium, actually both the epimers are present, but not in equal proportion. One may be higher, another one may be at the lower concentration. Example, you see, under prolonged storage condition containing, if a solution is have a drug, that is argometrine, This argometrin may undergo epimerization. For undergoing epimerization, it first of all need to be hydrolyzed, and then it get isomerized into argometrinine. Argometrinine isomerized into argometrinine. It's an example, right? The next one, it is the decarboxylation. You see the name itself, it's a decarboxylation. D means to remove, carboxy means carbon group, right? So elimination of the carbon dioxide from the compound, it is the decarboxylation. So the drug substance having a carboxylic group is sometimes susceptible for decarboxylation reaction. Example is your 4-amino salicylic acid. As it have salicylic acid have carbon, this carbon group, so carbon dioxide, actually CO2, so it has a tendency to undergo decarboxylation. The next one example, it is your procaine. Then the next one, next type of means process which will affect the drug stability is the elimination. So you see, in the elimination, what actually happened? Some group of the substance from the drug, suppose the drug have a structure, it have some functional group attached to it, right? Some group may get eliminated from that structure. If this group get eliminated from the structure, what will happen? It will lose, the drug will lose its property, right? Example, you see trimelamol, it's a drug. Trimelamol, it eliminates its hydroxymethyl group to form formaldehyde. Trimelamol, it actually eliminates its hydroxymethyl group from its structure to form formaldehyde. Clear? The next type of reaction, it is the oxidation. Oxidation, we know what is oxidation, right? So removal of a electropositive atom or radical or electron, or you can say addition of a electronegative atom, that is oxygen or radical. This process, it is known as oxidation. So the oxidation actually, it is controlled by the environment, which includes actually light, presence of light, presence of trace element, oxygen and oxidizing agent. So you see the conversion, the oxidation of epinephrine. How the epinephrine, it get oxidized, how these two hydroxyl group, it get oxidized to this. Clear? So thank you everyone for your patience hearing. If you like the video, just press the like icon. You can share this video and you can subscribe to my channel. Thank you once again.